Next, we will switch our attention to the cell cycle. The cell cycle is a series of changes a cell undergoes from the time it is formed until it reproduces. Two major periods of cell cycle include interphase and mitosis. During interphase, the cell grows and carries out its usual activities. In mitosis, the cell divides into two daughter cells. This figure shows the major phases of the cell cycle, which include interphase and mitosis. Interphase is a longer phase and divided into three subphases: G1, S, and G2 subphases. In the G1 phase, the cell undergoes vigorous growth and metabolism. Cells that permanently cease dividing are said to enter the G0 phase. During the S or synthesis subphase, DNA replication occurs, ensuring that the two future cells being created will receive identical copies of the genetic material. Without proper S phase, the mitotic phase will be incorrect. In the G2 subphase, the cell is preparing for division. Enzymes and other proteins needed for division are synthesized and moved to their proper sites. By the end of the G2 subphase, centriole replication is complete. The mitotic phase consists of mitosis and cytokinesis. Mitosis is division of the nucleus and divided into four subphases, namely prophase, metaphase, anaphase, and telophase. Cytokinesis is division of the cytoplasm and typically begins during late anaphase and is completed after mitosis ends. Before a cell can divide, its DNA must be replicated exactly so that identical copies of the cell's genes can be passed to each of the two resulting daughter cells. DNA replication occurs during the S phase. Replication occurs simultaneously on several chromatin threads and continues until all the DNA has been replicated. This figure illustrates the key components of DNA replication. In the first step, the DNA uncoils. Enzymes unwind the DNA double helix and expose the bases. The second step is separation. Hydrogen bonds between base pairs are broken and the DNA strands separate at the replication fork. The third step is assembly, where two new strands, a leading and lagging strand, are synthesized in opposite directions. The leading strand is synthesized towards the replication fork and the lagging strand is synthesized away from the replication fork. As you can notice, the old or parental strand acts as a template for synthesis of a new strand. DNA polymerase synthesizes a new strand on each parent strand, bonding free nucleotides together. After DNA replication is complete, two identical DNA molecules are formed from the original. The process is called semi-conservative replication because each new double-stranded DNA is composed of one old strand and one new strand. During DNA replication, histone proteins, which are made in the cytoplasm and imported into the nucleus, associate with the DNA, forming the two new chromatin strands. The two strands condense to form sister chromatids, united by a centromere. Sister chromatids are held together until anaphase of mitotic cell division, then distributed to daughter cells, ensuring each cell has identical genetic information. Now we will review each phase of the cell cycle individually. We start with interphase. During interphase, the cell carries out its normal metabolic activities and grows. The DNA containing material is in the form of chromatin. The nuclear envelope and one or more nuclei are intact and visible. Interphase can be divided into three periods: G1, S, and G2 phase. In the G1 phase, the centrioles begin replicating. In the S phase, DNA is replicated. In the G2 phase, the centrioles finish replicating and cells complete preparation for mitosis. Next, we review the mitotic or the M phase of the cell cycle. Most cells need to replicate continuously for growth or repair purposes. The mitotic phase is divided into two distinct events: mitosis and cytokinesis. Mitosis is division of the nucleus and is subdivided into four phases: prophase, metaphase, anaphase, and telophase. Cytokinesis is division of the cytoplasm. Together, mitosis and cytokinesis produce two identical daughter cells. We start with prophase. Prophase can be divided into two parts: early prophase and late prophase. In early prophase, chromatin condenses, forming visible chromosomes. Each duplicated chromosome consists of two identical threads called sister chromatids that are held together at the centromere. As the chromosomes appear, the nucleoli disappear and the two centrosomes separate from each other. The centrosomes act as a focal point for the growth of a microtubule assembly called the mitotic spindle. As the microtubules lengthen, they push the centrosomes towards opposite ends or poles of the cell. In late prophase, the nuclear envelope breaks apart. Special microtubules attach to the specific area on the centrosome called the kinetochore and serve to pull chromosomes to the center or equator of the cell. The remaining non-kinetochore microtubules push against each other, causing poles of the cells to move further apart. After prophase is metaphase. During metaphase, centromeres of chromosomes are precisely aligned at the cell's equator. The imaginary plane midway between poles is called the metaphase plate. Enzymes that will separate the sister chromatids from each other are triggered at the end of metaphase. After metaphase is anaphase. Anaphase is the shortest phase of mitosis. It starts abruptly as the centromeres of the chromosomes simultaneously split. Each chromatid now becomes a separate chromosome. Chromosomes are pulled towards their respective poles by motor proteins of kinetochores. One chromosome of each original pair goes to the opposite pole. After anaphase is telophase. Telophase begins when chromosome movement stops. Each set of chromosomes now at the opposite ends of the cell uncoil to form chromatin. New nuclear membranes form around each chromatin mass. Nucleoli reappear while spindles disappear. The next phase is cytokinesis. Cytokinesis begins during late anaphase and continues through mitosis. During cytokinesis, a contractile ring of actin microfilament forms a cleavage furrow. Eventually, this furrow pinches the cell apart, resulting in two daughter cells that are identical to each other and to the original parent cell. What are the factors that control cell division? Cell division is controlled by internal and external factors, which include the ratio of cell surface area to volume. This limits how big a cell can be and explains why most cells are microscopic. The second factor is chemical signals, such as growth factors released by other cells. And finally, availability of space. Normal cells stop proliferating when they begin touching other cells. This phenomenon is known as contact inhibition. Additionally, there are a number of checkpoints that occur throughout interphase. Checkpoints are key events in the cell cycle where cell division processes are checked and, if faulty, stopped until repairs are made. The cell cycle has G1 checkpoint in the G1 phase and G2M checkpoint between G2 and M phase. The G1 checkpoint, also called the restriction point, is the most important. If a cell does not pass the G1 checkpoint, it enters the G0 phase, in which no further cell division occurs.